Okay, good. So, hi everybody, again. My name is Adita, and I'm going to talk about MHC-I prediction tools. So I think Bjorn gave a very good introduction to all the tools in general, and now we will talk one by one and give maybe a little bit more detail. So this is basically a timeline, what I'm going to talk about. So I will give a short introduction to MHC molecules, even though most of you know quite a lot about that. Then I will present the user interface of our MHC-I prediction tool. And after that, we will also discuss a little bit of actually how to choose between all the different prediction methods that are available, and how do you choose a threshold to define what is a binder, what is not a binder, in order to select your potential epitopes. And then I will also talk a little bit about how do you choose between different HLE alleles if you're working on developing a vaccine, for example, that you want to work in a worldwide generation. So at the end, we will have a short exercise that you will have a chance to use the tool and try to use the tool and get to predict some malaria epitopes. So to start with, MHC-I molecules, most of you know this, but they are expressed on almost all nucleated cells and represent antigens to CD8-positive T cells that are cytotoxic T cells or killer T cells. And these molecules are closed, have a binding pocket which is closed at both ends, and that basically limits the length of the peptide that can be presented to 8 15 amino acids in length. MHC-I molecules, they are composed of one big alpha chain and a small beta-2 microglobulin chain. And this is actually the alpha chain, as you can see, is only this chain which is comprising the binding pocket. So on the alpha chain also is important for binding to the peptide. MHC molecules also are very highly polymorphic, so there are lots of different variants of these molecules that exist, all different proteins. And so that makes experimental characterization of peptide and MHC interactions a highly cost-intensive procedure. So that's where we actually need prediction methods. And for each prediction method, this was also shown by Bjorn, what we have is that to train or to build a prediction method, we need a peptide-binding data from experiments where we have a sequence of the peptide and associated binding affinity in IC50 values, associated binding affinity to a certain MHC molecule. And just to remind you, the IC50 values are basically concentrations, which the lower is the better score. And then we take some machine learning algorithms that we use, and based on this experimental data, we build a prediction tool. So as all the prediction methods are based on experimental data that we have, experimental peptide binding data to MHC molecules. So this is what we have so far, a data set. So we have MHC-I molecules for about 114 alleles covering. So, and this covers 80 human alleles, some macaque, chimpanzee, mouse, and others as well. This is from 2013, where all our predictors were actually retrained using this data set that we have. It's also important to mention that we cannot include all the possible binding data sometimes because we actually have to have a good coverage for each allele. So each allele has to have at least 50 data points in order to be included for the training. That's enough to get for the method to learn something. And you can find this data set online also if you would like to see that. And now I will introduce you our web user interface for MHC-I prediction tool. So you're also welcome to follow if you want, but later you will also have an exercise where you actually can try, and we will be here available to answer questions and if you have some problems as well. So this is the new website. If you see on the right-hand side, you will actually see that there is a section for T-cell epitope prediction. And under that, you will find MHC-I binding. So this is where the tool for MHC-I binding prediction is. If you press here, you will be sent to this page where you can see there is basically, it has several tabs, and I will go shortly through each of them. But this is the main submission page where you actually can paste in your sequence and do the prediction. But I want to go shortly through all of these steps because they are also common for other Allele methods. So the home one basically gives the main predictor. If you go under help, for each method you will also find a tutorial. So if you don't remember everything what we will tell you today, you can always find a tutorial where submission page is divided into several basically groups of steps, and each of the steps is also below this explained in details on what has to be done, what you can put it in, and other things like that. So that's a help page. Then you can also find an example page where you will find two small sets of peptides that have a predicted that were shown to bind to certain MHC alleles, and then you can use them and submit. If you press submit them, then you will basically be sent to the prediction method, and it will just show you that the method works. So this is just a small example that you can try to use in a prediction tool. We have also references, so a list of all references that were used, so basically papers that describe each method separately. And so these are the ones if you want to see how the method was actually developed and want to know more details about each method. And these are also the ones to cite if you're using the tool. There is also standalone packages available for different methods that you can download. And this is free from for nonprofit and academic users. And there are also licenses available for industry. So you can actually get to download and use a program on your own computer. And there is, of course, a help page where you can contact people from IEDB if you have any questions related to the tool. So this is a general, very general set for each of our methods that we have. And then I'm going to talk now about how do we actually use MHC+1 binding prediction tool. So we are back to the home page, the main page for MHC+1 binding prediction. And what we have on the top is where we actually submit our sequence of interest. So your protein sequence that you are looking potential epitopes in. And what happens is that you can submit sequence in different formats. So if you're submitting several, it can be space-separated sequences. Then it can also be just one continuous sequence if you wish. And then you can also have sequences in FASTA format, which is probably the most common format that you all know. What happens when you paste in your sequence here is that the method will break the sequence into short peptides of the length of your choice, and then you will have a full list of them and get binding predictions to MHC molecules. If you have many sequences and you don't want to copy paste, you can always just choose a file with your protein sequences and upload it here. The next step would be to choose the format of your sequence. So it actually is done for you automatically. There is an auto-detect format function. Basically the tool will identify what was the format of the sequence. So if this sign was used, then it will be treated as a FASTA sequence, and if this was not found, it will be treated as a continuous sequence. So there is one problem that might come up here is that if you had sequences separated by spaces, you would have to choose it here, because otherwise it would be treated as a one continuous sequence. So, and the next step would be to choose your prediction method. I'm going to talk about each of these methods a bit more in detail later, but we have several methods available, as you can see here. There is a list. And we also made it easier for you or for most of the users who don't know the details about each method that there is an IEDB recommended method, and this is the one we recommend to use. So basically that uses a consensus approach if the predictions for MHC molecules are available, and if not, then it uses MHC path. And I will talk in detail a little bit later about that. So after you choose your methods, you should also choose your host organisms. So you choose species from a drop-down menu here, and then you specify your MHC alleles of interest as well. and as well as the length of the peptide. So basically, what kinds of peptides you're looking for. Yes, so the question was that if for IEDB recommended method, is there is a downloadable package or it's just an online one? I'm not sure. Yes, there is a, okay, so you can, yeah, so you can get a standalone version for that as well. Yes, so when you choose alleles here and different peptide lengths that you want to get predictions for, there is also an option. If you click on here, you will find that you can actually, if you have many alleles that you want to get predictions for, then you probably don't want to choose them one by one. So you are also capable of uploading a file with all your alleles. So this should be in this format, that you have an allele written like this, then you have a comma and you specify a peptide length. So, and this can be as long as you wish. So you can get a long list of alleles uploaded at once as well. So after you chose all your molecules and sequences and alleles, then you also can choose how do you want to see your results or your output. So the output can be actually sorted if you would like to. You can sort it on basically on a score, based on a score, so that you get the best predicted peptide on top. Or you can just see your peptides just based on a position in a protein sequence that you uploaded it. Also, if you don't want to see all the results, but you just want to see some part of the results, depending on some threshold, you can choose to specify IC50 values cutoff if you want, or percentile rank score cutoff, and just see these results if you have too much too. So this is just choosing the output of the output format, so you can see a table, a HTML table, or you can get a text file as well. And you press submit. So this is it for the submission page. So this is what you have to take care of and select. And I just forget to mention one thing about choosing the allele. So if you notice here on the side, there is this statement saying that on the alleles that are only frequently occurring are shown. So this is for HLA alleles, and this is done in order to just have an easier usable user interface, because we have so many HLA alleles available, so that it becomes really huge. So by default, we are showing only the alleles that occur in a general human population more than 1%. So if you want to see some more rare alleles, then you will have to uncheck this box here. And then you will get a list of all possible alleles. So once you hit submit, you will be directed to this page where you can see your results. And on top of that, you will get your sequence or sequences. So this is just your protein sequence. The name will be used here if one was provided. And each of the sequences will get a unique identifier, which you can later see in your results table. And you can map it back and see where actually the peptide this sequence would come from. And in the actual result table, you will see what you get is the allele name, then you get a sequence identifier, then you get a position of the peptide where it started, where it ended, and the length of the peptide, the peptide sequence itself. And then you also see which method was used, if you chose IEDB recommended methods and not just the methods that you wanted to choose. So you will see which method was used, and then you will get a score. So here is a percentile rank score. And so what is it? So the percentile rank score basically is a percentage of randomly sampled peptides that are scoring better than your peptide. So how you do this is just that you take a huge amount, so that's 100,000 natural random peptides, and including the peptide of your interest. And when you get predict binding affinities to these, then we rank them based on binding affinity, so that the better binding affinity peptides go on top. And then we see where actually your peptides rank, and that's the percentile rank score. So that's just another score, which is universal, could be universal for all different methods. You can also get expanded result table where we will get more information. So here in this case, if we had several methods, so the consensus approach was used and it combines 3 different methods, then we can also, if we see the expanded result, we can also see basically what was the score for each separate method as well. And if you chose only one method of your choice at the beginning, then you will only get 1 score for that. And that will be in IC50 according to that. So as you see, there are a lot of different, or a few different methods that we have here in IEDB, and I just want to talk a little bit more about details of these methods so that you have an overview. So, and how do you actually choose the one that you have to use? So we can group prediction methods from HC+1 into 2 main classes, and that would be allele-specific methods and time-specific methods. So the main difference between these two is that when we have allele-specific methods, for training these methods, we use peptide sequences, and we are binding to one specific MHC molecule. So basically, each allele is trained at a time, one by one. And then we can get binding predictions only for the molecules that were part of the training set. You can, of course, predict new peptides, but you can only use a molecule that were part of the training set. While when we have time-specific methods, we have, for training, we have peptide sequences, we have binding affinities again, but we also include MHC sequence information into the training process so that the method can later extrapolate the new MHC sequences and can give predictions to any known MHC alleles as a sequence. So at IEDB, if we want to group these methods into these categories, we would have all the position-specific scoring matrix-based methods of SMM and the new version of SMM and combinatorial libraries. We would have here as allele-specific method because each allele is obtains one matrix, and then we would have artificial neural network MHC, which is called ANN at IEDB. Also here, and then also the consensus method, which combines all of these methods, would be in this category as well. While if we have some new molecules that were not part of our training set, then the NetMHC time method will be used. And that's a method based on artificial neural network. So these are the position scoring matrices, and Bjorn presented to you in detail how these are created and used. So basically, for each allele, we would obtain one matrix in our training set. We do the training set, and we would get like all 20 amino acids here, and then all peptide positions here, and basically each score will just say what's the impact of that amino acid being in that position. And these matrices, they can be obtained in different ways. So we have several methods that do it in a bit different way. So we have an SMM method that just uses ridge regression to minimize the difference between predicted and actual measured binding affinities or binding scores. Then the new SMM version also includes a Bayesian prior, which prioritizes these matrices that include some amino acid similarities into that. And then we have combinatorial library methods, which basically creates, based on making huge combinatorial libraries where one position is kept constant and is varying different amino acids. One amino acid is kept constant and all the other amino acids are varying. So, but the details of these can be found in papers if you want, so we can discuss this later on for those who are interested. Another group of methods is based on artificial neural network, and that's a machine learning technique, which is works very similar to how our brain is actually working. So it's capable, neural networks are capable to capture nonlinear interactions between input and output, and it's also capable on generalizing to the new examples after the training process. And these are NetMHC and NetMHC time methods. So NetMHC, as mentioned before, is an allele-specific method. So it can only give predictions for a chosen for the real that was part of the training set. And MHC-PAN is a pan-specific method which can actually give predictions to all possible MHC molecules, and that's because it includes the information about MHC sequence. And that's done in a format of so-called pseudo-sequence, which is basically composed of the residues at positions that are important for peptide binding within a peptide binding process. And then we have a consensus approach, which was created based on observations that separate combining the different methods can at the end give improved predictions on each of the methods by itself. So in this case, in IEDB, consensus method is combining MHC, SMM, and combinatorial libraries methods. So these are allele-specific methods. And in order to combine different methods, we have to have a unified score, which, for example, combinatorial libraries doesn't measure IC50 affinity score. So, but in all of the cases, we can get a per sample rank score, which I just presented before. So this is what the consensus approach is using. And if we get back to our expanding result table and just take one result entry, for example, then we can see all the different, so we can see that consensus method was used, and it combined the ANN and combinatorial libraries and SMM methods, and we can see all different scores for these methods. So what ANN gave, SMM, and combinatorial library, and then we have a score for each different method, and basically the consensus method will take MEDN out of these. And that will be 0.4, and you can see that the result is 0.4 here as well. So this is how the consensus score is calculated. So our recommendations for the users would be use IEDB recommended because that basically addresses the problem for you. So what it does, it basically takes your molecule of interest and it will choose the best method for you. And this is done, so as mentioned before, this uses consensus if we have these molecules in a training set, and if not, it will use MHC-PAN. And this is based on benchmark studies, the IEDB recommendations, and you just have to keep in mind that once we have new benchmark studies coming up with more data, these recommendations might change as well. So how do you know predictions are available for certain molecules or not? Well, you don't really have to know that. I mean, the method is chosen itself because we know it. But the list of alleles for the method we're trained on, is it available publicly somewhere?
But you're using the web version.
Yeah, the method will choose for you.
Yeah, you'll be.
So you don't need to worry about that because we know and the method knows and the method will choose the best prediction for you. So if it found a molecule in a list of training molecules, it will use a consensus approach, and if it didn't, then it will use an MHC.
And the other way, so if you're using the web version, if it's not on the drop-down list, we don't have the algorithm. So basically, you can check.
I mean, in general, if you cannot give any predictions at all,
yeah,
then it will be not on the list.
But in the admission time, just choose.
Yeah, if you know what you're doing, I mean, if you know, then you can choose your method as well. Yes.
Usually.
Then, I mean, you don't have to if you, so IEDB recommended this for the users that don't know all the details about the method and when it's good and if it's good for your molecule, how it's going to work. So then we could just use IEDB recommended and it will choose for you. Yes?
What if you're running standalone version? Get the list of what we need to method system.
Yeah, so the question was if you have a standalone version, can you have a list of alleles that the methods are trained on? This I'm not sure. You're? Yes? Okay, so the standalone method will provide an option that shows the list of alleles that are trained on.
No, if you specifically want to know what all alleles are there, if there's a command, we which you get the complete list of alleles. But it's not given
So
publicly.
that's a generic one. So that is for each allele as well?
Each method and
Okay.
each all alleles are available for each method. But basically,
Okay, so each method
the
will have-
easiest method to test is just use the allele that you want to know if an algorithm does exist or not. Just type the command in there, it will do the prediction.
No, but that's for what the method was trained on. So not for what it can give the prediction for.
You're asking?
No, but that makes it time can
Yeah.
yeah. Yeah, for allele-specific, yeah. So the lists are available.
Yeah.
That's okay. Yeah. Okay. And this doesn't work. Okay, so now we talked on how to choose a prediction method, and another problem is actually how do you choose a prediction threshold to define binders. So there are two main approaches on how can you actually define a binder, and there are studies that show that how this works. So the most frequently used is just IC50 affinity threshold, which is 500 nanomolar, which was shown to actually identify binders that are also recognized by T-cells. If you really want the high affinity and would take some strong binders, you would use 500 nanomolar, and 5,000 nanomolar would actually correspond to quite low affinity. Another approach would be to take top 1% of the peptide for all your MHC and peptide length combinations, and then in this case, you would cover most of the immune responses. And there are studies also that investigated that experimentally. And so if you use the first approach, what you will get is that you will make sure that all your peptides have reasonable binding affinity, while if you use the second approach, which should be second here, then you will make sure that you have an equal number of peptides for each allele. So there's also a new study by our group, so that's made by Timo and the others in the lab, that shows actually that the size of the repertoire size of the binders for each allele can vary. So if we have a set of peptides that we want to check how many of these will be binders to this allele, then different alleles would have different number of binders from the same set of peptides. So this was also shown in the study that for using dengue virus epitopes. So if you peptides, basically, if you just check what's the fraction of peptides that are binding at IC50 threshold of 500, you would get different percentages for different alleles. So the repertoire sizes vary quite a lot for different MHC molecules. And also, it was shown that if you want to predict, for each allele, if you want to predict 75% of epitopes at IEDB, you would also need a different threshold to predict that. So basically, if the allele has a high binders repertoire, it would be also associated with an epitope with higher affinities as well. So we can say here it has higher repertoire that it can bind, while compared to A101, and then it also has lower affinities for epitopes as well. So this gives us an idea that there might be allele-specific affinity cutoffs that we have to use, because alleles are just binding different repertoires of peptides and might have different affinities. So this was also done by people in our lab, so that you have a list. So here, if you come back to the help page of the tool, you will have actually a list of these suggested affinity cutoffs for different alleles as well. So you can use that also. So this is a new study. So in general, we would recommend to combine affinity and ranking scores, so that you choose IC50 lower than 500, and then you can also consider to take top 1% of the peptide, as mentioned earlier. But you should also keep in mind that there are studies now that are showing that there should be allele-specific threshold, and that this should be a good idea to use as well. Yes, sir. So the question is if there was a study done to compare self-antigen affinity and basically peptide compatibility. Did it happen? No. So we don't know that. But yeah, that could be a good idea. Yeah, so the next step is also how do you actually choose the alleles, right? So if you're working on a general problem where you're trying to create a vaccination that would work in maybe in a certain region or in a general human population, we have to consider, as I mentioned before, MHC molecules are highly polymorphic. So we have more than 6,000 HLA of different alleles existing in humans, for example. And if you remember the number I gave you before, we have binding data only for 80 of these. And that's a very, very low percentage that we have binding data for. So how do we deal with this polymorphism? And which alleles do we actually choose if we want to create a vaccine that works worldwide? So one good thing is that actually it's known that the frequencies of different alleles are different across the world. So there are different populations that have some alleles are more common than the others. So if you have, for example, if you take the 8 allele and in Japan there will be 0% of the population that has this allele, while if you go all the way to Brazil, that will be 11% there, and for example 15% in Russia. So the alleles basically have different spread across different populations, and these frequencies can be found here if you're interested in that. Also, there were studies by people here at the Institute in Sydney that showed that MHC alleles, they also share some binding similarities. So they can actually be kind of classified into few supertypes because they still have some similarities. So taking all of this into account, we obtained a reference set of 27 alleles that together would cover 97% of the human population. And these are the alleles for HLA and HLA-B, so alleles for human, and these are the worldwide frequencies of these alleles. And this table is also available. Basically, you can use this set of reference alleles very easily. So if we get back to the submission page, where you were choosing alleles, if you remember, there is a link here which you can press, and you will get this select HLA allele reference set, and then you will get all the reference sets for you. So that's an easy way to do predictions for all these common 27 alleles, and that chooses also different peptide lengths for you. So that's a quick way to get that. So now, if you don't have more, or do you have more questions to this? Because now we're going to move to the exercise, where you can actually try. So if you have your computers, all of you, it would be good if you can just get them out now, because we're going to try to use the prediction methods and try to find the malaria epitope. So the exercise is based on this study down here, which you can also go to PubMed and get. So it's based on this AMA1 protein, which is expressed in the membrane of Plasmodium falciparum, and it has been tested to give CD4 and CD8 positive responses, and it's considered actually to be a good candidate for malaria vaccine. So in this study, they had 5 volunteers that were immunized with this protein, and then the protein was scanned for all 15 years. And then there were T-cell responses basically were measured, and for these different 15 years in different volunteers, and volunteers were also HLA-5 figures, were sets of HLA molecules. So the problem statement is here, and I will leave this open, but I also think you should have an email with the sequences so that you can copy paste them. So what you have to do is you have to use IEDB prediction tool, which I just presented, and to find minimal epitopes of the best epitope within this 15 there. And you also know volunteers' HLA allele set. So now if you can try this for about 15 minutes, and I will be here and other people, if you have any question or if something doesn't work for you on the slide, you can answer that. So 15 minutes for the exercise. And I'm just going to go through very quickly with the answers. So what you had to do basically is, this is the solution, you had to take this 15-layer sequence where you know that there was a response in the donor, so you know that the epitope is somewhere in this sequence. So you take this sequence and then you just leave them out of the text format. You just use the IEDB recommended in this case, so you don't really just think about it. And then here you had to choose a list of alleles that the donor had. And as we don't know actually how long was the peptide which was giving or the epitope which was actually giving a response, then we just choose all lengths. And the method will scan for you for all possible peptides within the 15 there. And we can also choose to sort peptides in the output based on percentile rank score so that we get the best epitope or the best peptide binder on top of the list. So when you press submit, the results should look like this. Everybody managed to get here. And you can see that on top, we have this peptide, which gave a percentile rank of 0.2. Okay, and there was a question here before in the audience, so what would be considered to be a good percentile rank when we choose strong binders, and that would be about 1%. So if everybody managed to get this, then it's very good. So you're good to use the prediction tool here. And just to show you that this peptide here was actually shown to give a T-cell response in the study as well. So if you would like to, you can read this study.
This would be restricted to those people who are HLA-B1801, or we generalize to other alleles. Everybody's B1801.
Yes, not everybody. So the question is if this would be restricted to HLA-B1801. So in this case it is. So this peptide is giving a strong prediction to HLA-B1801, but so if you want to use this for general vaccine, that's a bit more detailed that you probably have to discuss with your.
Are you done with it?
Yeah, I'm done. Actually.
Let me just say something about that.
Yeah, let's talk. Okay. So I think some important points here are, and I guess that's useful here. So as oops, wrong one. 1% is our cutoff, meaning there's 3 peptides here, right?
Yeah.
That are possible. And so this peptide is predicted to bind the top less than 1% for B1801, and it's predicted to bind B4402 and it's the exact same peptide. So actually, in this type of experiment, we don't even know for sure if we test it in that patient because he has both alleles, is it going to be restricted to one or the other? And you need to use something like a single HLA transfected cell lines or something like that to really clarify it. But so this is actually a typical outcome if you look at it. And then there's another one that could be, right? This is another possible restriction. And then potentially, I mean, after we go up to 2% or something, this is another potential one. So there's always multiple possibilities. So obviously, but if you would have to choose one, totally I would go for this one, because also there's two shots, you have 2 different alleles where it can bind to, and both of them are high affinity, yeah. And as we concluded, it actually works. So, but again, in this example, this was in the real study. So essentially, they had ordered 15 mirrors and scanned them in these patients, and then find the activity, and couldn't really say what is actually being recognized. So that's when they synthesized these peptides based on the predictions, and then had actually very good success. And the nice thing is also, I mean, you can use these 15 mirrors and just throw them in there, and the T cell will kind of manage to typically they get cleaved because you have proteases in your cultures, and they will be cleaved together, and they will somehow fit in. But if you use exactly the right peptide, you get 10-fold or 100-fold higher responses because you're really stimulating the protein exactly to be recognized. That's a big benefit of working with the exact peptide. I guess that was, that specific donor has.
So if you take this particular bacteria, and then put it into the epitope search and find all the other epitopes that can respond to this, then probably we may cover that one.
Right. Right. So these are different applications. Right? In this case, again, it's a real case where they still had blood left over from the same donor and wanted to then to see if that specific donor was. That's exactly what you're saying. It's the right way of to generalize it. Yeah. Okay. So.
Yeah. Thank you. And so the people have more detailed questions about the different methods. So we're going to talk about this in one session, the breakout session. Yeah. So I would be happy to discuss that now. Thank you.
Let's take a break.
Let's take a break.
15 minutes.
15 minutes break.
Yeah, let's just 20 minutes. 20-minute break.
20 minutes break.